in the area of human and animal reproduction. He now serves as Chief Scientific Officer at Boston IVF and as an Associate Professor at the Yale University School of Medicine. Dr. Sakkas' topic is live imaging of human embryos, what lies before the surface. Dr. Sakkas, welcome.
Thanks, Billy. That was a bit long. So today I'm going to talk about something that I love watching. I love looking at embryos. I think a lot of the embryologists here like looking at embryos. But I'm going to talk about what lies below the surface and some of the tricks the embryo uses to actually fool us. So today I'll talk about current methods of embryo imaging. Hopefully help you understand what live imaging tells us about the pre-implantation embryo, discover how the embryo can fool us, and examine some future technologies, hopefully at the end, that will allow embryo selection using live imaging. For this talk, I don't have any conflicts of interest. So, we know a lot about pre-implantation embryos, and if you look at I don't think I've got a laser on this, but if you look at the top, when we look at Xenopus and Drosophila, we talk about hours of development, but then when we look at mouse, bovine, and the human in particular, which interests us, we're talking about days of development. And even within days, there are a lot of differences between these embryos. And in general, what we look at as an example for human is a lot of times the mouse embryo, but there are a lot of differences. And then we have different tools, so we can use invasive tools, biopsy, as Santi, has professed for many, many years. We use micronuclear injection, to inject RNA, and we can track these. But there are many non-invasive techniques that we can use in embryos because we have amazing availability to embryos. We can look at the culture media, we can image the embryos, we can look at output, uptake, production, we can look at many things, and we can actually live image them, and we have many of these techniques. One of the techniques we use is also fluorescent dyes, and on the left there are fluorescent dyes that you can use and analyse embryos when you fix them, but actually I'll talk also about some new technologies using what are called spy fluorochromes that we can actually image embryos live, and we can actually add these fluorescent dyes, and they're not harmful to the embryo. And these are techniques we used many, many years ago, but these are actually more up-to-date fluorochromes. So there are many ways we can learn about human embryos. I don't know if this will work, probably won't work. So time-lapse, as we know, is super impressive, but it can only tell us so much. And if you look at the mouse on the left and the human embryo on the right, if the video worked, you would see that even though they behave very similarly in cleavage patterns, and we learn a lot from them, you see a lot of different timing in the development of the mouse versus the human. Obviously, the size is different, but now we're using time-lapse imaging for with artificial intelligence, which I'm sure you'll hear a bit later. But time-lapse still gives us a little bit of a black box, about what's actually going underneath the skirts of the embryo. So time-lapse is super impressive, but it really can only tell us so much. There's some other technologies we've been working on for many years. We've been doing this for a long time. And some of the things we've been interested in and we can look at non-invasively are things like the metabolism of the embryo. And we've put a lot of work into this over many years. This is just 2 papers that we had published using a technology called fluorescence lifetime imaging microscopy. This technology, it's not just looking at the brightness of the embryo, it's actually getting much more information using fluorescence. So the clue is in the lifetime imaging, whereas when a pulse is sent by the laser, you can actually measure the time it takes to come back and excite the actual photons. And this technology is really cool because what it does, it allows us to non-invasively look at the metabolism of embryos. Obviously a lot of other cells. And on the left-hand side is NADH, and on the right-hand side is FAD. If you vaguely remember any of your biochemistry, these are very important parameters in mitochondrial metabolism. And you can see here on the left-hand side, you can see actually that the embryos actually get brighter as they get to the compacted stage. and then they decrease in brightness. And the FAD actually, if I go back and play this again, you can see the embryos get really brighter at the compacted stage and then decrease on the left-hand side in brightness, whereas on the right-hand side, the FAD actually increases in brightness. And we get a lot of information from using this technology. We've learned actually a lot about human embryos, things that we sort of knew, but this technology validated it using live imaging. One of the first things we saw was that that human blastocysts show very distinct metabolic signatures between the inner cell mass and the trophectoderm. And if you look on the graph on the bottom left-hand side, you can see the inner cell mass are the little blue circles, and the orange are the trophectoderm. And these are from the same embryo, so you can compare one embryo with another. And using some of the parameters that we measure using fluorescence lifetime imaging that indicate mitochondrial metabolism, you can virtually draw a line between the blue and the orange, showing that they're very distinct in the way they metabolize. So this is important because the inner cell mass and trophectoderm cells have very different metabolic states. The other thing we're obviously interested in, is metabolism the same in a really beautiful embryo versus a bad embryo, or two really beautiful embryos? And what we found when we plotted blastocyst morphology versus metabolism, not really surprising to the geeks of us that are sort of metabolic embryo specialists, but actually metabolism really didn't have much to do with actually if you were a 3AA or a 3AA or 3BB. So morphology and metabolism gave us very different information, independent information. It wasn't synergistic. The one I really liked, being sort of loving embryos and like looking at them, is when we actually looked at the metabolic state of day 5 embryos and day 6 embryos, regardless of their morphology and what stage they were, we found that the actual embryo, again, when we looked at certain parameters, and you can see the yellow ones in the circle, the circles with yellow are day 5 embryos, the darker circles are day 6 embryos. We found that the embryo actually has like an inside clock. It has its own circadian clock rhythm that knows it's day 5 and it knows it's at day 6. So this is fascinating when we talk about day 5 and day 6 embryos. Even though it was at different stages or different morphologically, day 5 and day 6 embryos are very different in what they know they're doing metabolically. This is a really old paper, but it's a very interesting paper. It was published by Michelle Lane, who unfortunately passed away several years ago, and David Gardner. And what they did, they measured the metabolism of all these mouse blastocysts that looked exactly the same. So they had exactly the same morphology. And on the left-hand side, you can see their glycolytic activity, so their glucose uptake, was a high range. And what they did, they took the ones on the left-hand side, which were in the sort of lower 15% glycolytic activity, and transferred them, and took the ones on the right-hand side and transferred them. And they found that the ones on the left-hand side, which had a more innate glycolytic activity, had a much higher viability than the ones that actually had the poor metabolism. And again, I'm talking about embryos that you couldn't distinguish them morphologically. So we've known this for many years. So we thought, okay, we've got a really cool technology with metabolic imaging using fluorescence lifetime imaging. We wanted to see if we could measure parameters in embryos and distinguish them metabolically to see if we could pick which ones are the viable embryos and which ones are the non-viable embryos. So we performed this pilot trial. We consented 121 patients. All of them had more than one frozen embryo, so these were good human blastocysts. What we would do, we would actually consent the patient. The embryo would be thawed, so all the decisions were not based on the metabolism of the embryo. They were already sort of designated because they were good morphology embryos. We measured, once we thawed the embryo, it took us about 6 minutes to measure the metabolism of these embryos using the FLIM technology, and then we transferred it back to the patient. And what we had then, we had the metabolic profile of the embryos that were transferred back to the patient. And we could then distinguish, the aim was to distinguish whether we could pick which embryos were viable using their metabolism, using the ones that were not viable. So what happened? When we looked at the logistic regression, looking at all the metabolic parameters we had, and these are mitochondrial metabolic parameters, we were unable to improve the accuracy of actually picking embryos, or blastocysts at least, good blastocysts, and predicting them. We could get, the highest we got to was about 0.62, so 62%, but that was not enough to compare basically against, good morphology. So unfortunately, even though we could do a lot of, we learnt a lot about embryos, we actually failed to use this metabolic parameter to pick which ones were viable or not viable. What was interesting though, out of all these data sets, we could still see various things. We could still tell you which blastocyst was day 5, which blastocyst was day 6. We could tell you with nearly 90% accuracy what the inner cell mass was and what the trophectoderm was. We could tell you the grade, we could tell you the age. What we couldn't tell you was which one would lead to a pregnancy, unfortunately. The other thing that was really interesting that we could tell, with 80% efficiency, we could tell you which one was euploid and which one was aneuploid. So it's interesting. We could tell a lot of things about these embryos, but we still couldn't tell you which one would get pregnant according to its mitochondrial metabolism. So why did the FLIM fail to predict viability? It could do so many things, tell us so many things about embryos. Well, if you go back to some of the old papers, and we probably should have read these about 10 years ago before we started all the FLIM work, eggs human eggs or mammalian eggs and embryos are very particular. They're really interesting. So as we all know, and Johan will probably tell you later about the oocyte, it comes with this huge packet of mitochondria. But these mitochondria are very specific to oocytes. They're a different shape. They actually, at the beginning, there's hundreds of thousands of them, and then they actually decrease in the amount. So what we're looking at is the oocyte mitochondria, if you want. They decrease, they decrease, they decrease, and around the late blastocyst stage, during peri-implantation, there's actually a switch, and you start getting biosynthesis of new mitochondria, which is what takes part of our body, what we're all walking around with, basically. So the mitochondria we measure in the embryo, in the end, is very different from what is actually going on post-implantation. So what we were probably measuring is the good mitochondria that actually gets you to the blastocyst stage. But it wasn't necessarily representative of what's going to get you post-implantation. So we learnt a lot during these, but there are still some really cool technologies using non-invasive imaging that don't just look at the mitochondria, but can look at many things. So one thing I think I failed to measure is that when we're measuring with FLIM and NADPH and FAD, do what's called autofluoresce. So we're not adding any fluorescence tags or anything to these embryos. But there are many things in our bodies, proteins, other factors that actually autofluoresce and are excited when you actually image them with laser. And hyperspectral microscopy is actually one way of measuring multiple factors rather than focusing just on the mitochondrial one. So this data by Jeremy Thompson group and others is actually being published now, and Kylie Dunning now has actually used this technology to distinguish between aneuploid and euploid cells in mouse embryos using this non-invasive fluorescent technology, which is very promising, I think. Time-lapse and FLIMs are impressive, but there are technologies now that are way better at actually looking at imaging embryos. And we've been lucky. Many years ago, I ran into a guy called Nicholas Plachta, who's a brilliant imaging specialist around the world. He gives fantastic talks, so if there's someone you want to invite, that's someone you can give. And Nico and we collaborated on this paper, and we used these spy fluorochromes, which we could use for live imaging, to image live embryos, to image human embryos live and blastocysts. And we found that these fluorescent dyes are actually not harmful to the embryo. We did a lot of testing with mouse and got pups and everything, so it worked. We see that live imaging reveals very interesting characteristics of human blastocysts. We found that blastocyst expansion causes trophectoderm to do really interesting things. And obviously we also saw that we also were able to see cytoplasmic DNA being extruded by embryos live. So this is an example of a hatching blastocyst. It's fascinating how the trophectoderm, they're in such a concert, and how the actin filaments, so the blue here, and you'll see in the following slides, the blue is actually the nucleus, the red is the actin filaments, but you can see how the actin filaments really reach out like neuron cells to actually assist in hatching. And we can do time lapse of blastocyst hatching. We see an amazing concert of activity to sort of push out of the zona, and how important the shape of cells is at the trophectoderm, and how important the actual actin is in actually pressurizing the zona pellucida to actually help the embryo hatch out. So we're learning a lot of things, and obviously, you know, the cool thing we can look at is mitosis, and you can, if you track mitosis, you could actually see very interesting things. So, you know, we can obviously follow the beautiful chromosomes missing, but if you look at the one on the last one on the second, the top second right, you can actually also see some actually chromosomes lagging. And, you know, obviously we know how embryos are bad at actually being euploid sometimes. But, you know, we can actually visualize chromosomes actually lagging when we look at these technologies. And one of the interesting things we saw, which maybe is not that surprising, but we saw in the bottom right-hand side, you can see the mouse duration of mitosis is much tighter compared to human. And that we also see with time-lapse. So we're learning a lot with these. We also are getting a really good understanding of how you allocate to inner cell mass and how you allocate to trophectoderm. We can, you know, the old theories of Martin Johnson and that, we can validate a lot of those, but we can look at these more at the molecular level, where you can see that inside cells do actually, you know, have a propensity to go more to the inner cell mass than the outer cell. So we're learning a lot about cell positioning also. If you look again at this blastocyst, we did a lot of blastocysts, and we started seeing some interesting things. So you can see in the arrows there are actually cells that are actually putting out these nuclear buds. And we've seen that quite frequently, and you can also see the hatching happening here in these blastocysts. So we saw that blastocyst formation is really dependent on hatching, so we can follow mitosis, we can follow positioning of trophectoderm cells, we can follow the different sort of stages of mitosis in all the cells and see do inner cell mass differ to trophectoderm. So we've learned a huge amount of information from these embryos. I showed you the video before the blastocyst, and if you spend enough time looking, and it wasn't me, but it was some of the collaborators we had, you could actually start seeing that some of these cells were putting out these nuclear buds. And this was actually fascinating to us, because at the same time we were obviously interested in non-invasive PGT. And one of the questions people have is, you know, why is this rubbish? What are you finding? Why are embryos putting out DNA into the media? The whole, Santi showed us the slide of why it's happening, but what we definitely see is that blastocysts do this on purpose. And what we do know, it's not related always to apoptosis. So we could double label these embryos with apoptotic markers. We see that the embryos are doing nuclear budding for some reason. We still don't understand why. My favourite theory is that it's sending a message to the uterus, to the epithelium. We know that happens in other species. So this is all part of what I'll conclude of, and I'll talk a little bit about later. But we can actually track the DNA being secreted by the nucleus, and we can actually follow them, and you can follow DNA being actually pushed out in nuclear buds, and we can track these and then actually see what's actually causing it. And I'll briefly just go over some of the things that are causing it, why nuclear budding happens. We know that it's cells that are actually normally in the trophectoderm, and it's like a squeezing phenomenon. So the cells on the trophectoderm that are being squeezed, they're the ones that are actually putting out more of the of these cytoplasmic buds. We also know that it's more likely to happen when there's not what's called, when there's not a keratin cage around the cell. So keratin, one of the filaments that we know is very important in actually the shaping the embryo, when there's a cage of keratin around the cell, it's much less likely to happen. So when it's not there, it's actually, the cells will actually secrete cytoplasmic budding. The other thing is what we found is actually biopsy triggers DNA shedding. So when you're biopsying an embryo, you're also triggering this event because you're actually squeezing those cells, the trifectoderm cells, and there's more cytoplasmic DNA being extruded. And this is just showing on the right-hand side, you can see cells with more of a cage of keratin around them are much less likely to secrete or to have nuclear budding. These technologies are amazing, and just a few months ago, and I know some collaborators are here, Magdalena and the groups published using similar technologies showing that even the first two blastomeres, so the two-cell embryo, contributes unequally to the human embryo. So there's certain knowledge in the first two cells that actually will guide you to becoming inner cell mass or trifectoderm or epiblast. So it's amazing what the embryo can do. So there are a lot of tricks used by the human embryo to fool us. And we even know, and it's sort of controversial in some ways, there's possibly depletion of aneuploid cells in human embryos and gastroids. So there's some knowledge in the embryo that it actually can possibly weed out aneuploid cells, and that's actually been shown also by extended in vitro culture of human embryos, where you can track aneuploidy of embryos that have been diagnosed as aneuploidy, and when you do extended culture, some of those aneuploidies disappear. So the embryo has a lot of tricks. So diagnosing the blastocyst is interesting, you know, when we're doing these techniques. Millions of years of evolution may be smarter than 20, 30 years of molecular biology. So we have to really be careful when we're doing these diagnostic techniques, and Santi's right, PGT is an amazing technique, but the embryo has a lot of tricks up its sleeve to fool us, I think, because the aim of every human embryo is to have a baby. So it has a lot of things that it wants to do to survive. It can correct aneuploidy maybe, that's controversial. Budding of nuclear DNA happens, and it's not apoptosis all the time, and it happens most of all in blastocysts, and that's why when you do non-invasive PGT, if you're doing it before day four, I wouldn't even worry about it because it's just rubbish DNA. But day 5 and day 6, it's probably more important. There's a metabolic switch that knows the embryo is at certain stages. It knows which cells are functioning. It's really clever in doing these things. Humans are one of the only species that will do ectopic implantation. So a blastocyst wants to implant somewhere. And the blastocyst and the human ignore the implantation window. There is no other species in the world that you can put a 2-cell or a 4-cell or a pronucleus in the uterus and get a pregnancy. So when Bob Edwards and Steptoe did the first transfers, if they did that in any other species in the world, it wouldn't have worked and we wouldn't be having this conference here. There's preferential cell contribution, as we saw from the 2-cell, and there's much more the embryo can do. So embryos are very, very clever. And all of these tricks are there to maximise its ability to have a live birth. So maybe in the future we'll be doing different confocal microscopy, we'll have different markers. We could probably do this now under light microscopy. There are technologies out there. So I think imaging of embryos in the future will be very important and will be very different from how we do it now using embryoscopes or the Jerrys. So it'll be very different. I'm lucky I get to come to San Diego and give these talks, but there's a lot of people much smarter than me behind these technologies, including the people at the top who were involved with the FLIM. The crafty-looking guy on the bottom left there is Nicholas Blachta, probably from looking at too many embryos under fluorescence, and obviously the team at Boston IVF, which do all the baseline work for us. So I'm sorry I've gone a little bit over time, but if you have any questions, feel free to ask me or email me in the future. Thank you.